Hi, my name is Kim Fake, and I'm a wildlife research coordinator at the Urban Wildlife Institute at the Lincoln Park Zoo. Today I'll be talking about our study evaluating small mammal community composition and health across the Chicago metropolitan area. Small mammals play an important role in urban ecosystems due to their top-down effects on vegetation and bottom-up effects on predators. Yet little is known about how urbanization affects small mammal communities in terms of their community composition and their health. So urban environments pose unique challenges for small mammals, including excess sound, light pollution, landscapes dominated by non-native plants and impervious surfaces, fragmented habitats, among many others. Some species may not be able to survive in such environments, and some may simply tolerate or adapt to them, while others may thrive in urban environments. But little is known about how urban environments affect the composition of small mammal communities. Challenges in urban environments may also have detrimental physiological effects. Under non-stressed conditions, the hypothalamic pituitary adrenal axis, or the HPA axis, produces glucocorticoids, which are stress-associated hormones. These stress-associated hormones are produced as a normal part of homeostasis and at baseline levels. These baseline levels depend on the species, its age, its sex, its reproductive status, and the season. However, in urban environments, small mammals are exposed to environmental stressors which can affect the HPA axis, causing it to produce more glucocorticoids above baseline levels. So urban environments include potential stressors such as human activity and presence. There's unique habitats dominated by non-native plants and impervious surfaces, which could affect the ability of the small mammals to find cover from predators or access to food resources. Small fragmented urban habitats or altered abundance and diversity of small mammals in urban spaces may also affect competition. So all of these stressors can potentially cause the level of the glucocorticoids to rise above baseline levels, and chronically elevated stress-associated hormone levels can lead to immunosuppression and has been linked to decreased survival. So furthermore, differences in community composition and environmental factors can have cascading effects on the dynamics of hosts and their parasites. So some parasites may disappear from cities due to lack of hosts available to support their life cycles. Alternatively, populations of urban mammals may occur at higher densities and forage in areas of clumped food resources, causing increased exposure to or transmission of parasites. So ultimately, with this study, we hope to determine how small mammals respond to urbanization in the Chicago metropolitan area in terms of their community composition, abundance, survival, stress levels, and parasitic loads. Today, I'll touch briefly on the effects to community composition, but I'll focus mainly on the effects on stress levels and parasitic loads. So for this study, we chose 10 study sites along an urban gradient in the Chicago metropolitan area. And our 10 sites consisted of 4 urban sites, 3 ex-urban sites, and 3 intermediary sites, which we refer to as transition sites. At each site, we set up a 15-meter-long vegetation sampling transect. Along this transect, we conducted a line point intercept method to estimate the percent native vegetation and the percent ground litter cover. So we conducted trapping in the summer months of May, June, and August in 2017, 2018, and 2019 for a total of 3 trapping sessions per year over 3 years. And each trapping session would consist of 3 consecutive trapping nights. So we would trap small mammals using Sherman traps, and the traps were set up along 4 100-meter long transects at each site. And then for each small mammal we captured, we would record its species, its age, sex, reproductive status, and its body weight. So for each small mammal, we also inspected it for any ectoparasites and recorded any that were present. If there were any ticks present, we would remove those and place them in vials of 70% ethanol. And we also collected a small hair sample from the hindquarters of the small mammal and placed that into paper envelopes. The hair samples were returned to the Lincoln Park Zoo endocrinology lab, where the glucocorticoid metabolites, essentially the stress-associated hormones, were successfully extracted from the hair using 90% methanol and then quantified using an enzyme-linked immunoabsorbent assay. Over the course of the study, we captured 909 individuals consisting of 11 species. Meadow vole and deer mouse were the two most commonly captured species. So first we conducted a permanova to see if the site type, essentially the urban intensity, had an effect on community composition of the small mammals. And what we found was that community composition was significantly related to site type. So here I have an NMDS plot. So basically in this plot, each symbol represents a particular site during a particular year and trapping season. The exurban sites are represented by the pink squares, the transition sites are represented by the green circles, and the urban sites are represented by the blue triangles. So the closer together two of these symbols are, the more similar their small mammal communities were. And as you can see that we see a bit of clumping of the different site types together, indicating that the sites of the same site type had a similar small mammal community. As you would expect, some species were found more in urban sites, while others were found more in exurban sites. So, for example, we trapped more house mice and Norway rats in more urban sites, while we tended to capture least shrews and northern short-tailed shrews in exurban sites. So next we examined the stress-associated hormone levels of meadow voles and deer mice, as these were the two most commonly trapped species, and they were also found in all three site types. So when we initially looked at stress-associated hormone levels, we started by just looking at the two species, meadow vole and deer mouse, separated by sex and site type. So here in this figure, you can see we're looking at the stress-associated hormone levels, the cortisol levels of meadow voles, and we have separated them with the males on the right and the females on the left. The pink bars represent the stress levels at urban sites, the green bars at transition sites, and the blue bars at exurban sites. So as you can see here, the meadow voles trended towards lower stress levels in urban environments compared to exurban and transition sites. But if we look at the deer mouse, we actually saw the opposite trend. So deer mice tended towards higher stress levels at urban sites compared to exurban and transition sites. However, in this analysis, we didn't take into consideration the non-independence of individuals sampled within the same site or transect. And also, as I previously mentioned, community composition and habitat characteristics may also affect the stress-associated hormone levels. Furthermore, here we're just separating the different sites by site type as a way to measure urban intensity. But this is a very rough kind of high-level measure of urban intensity then. And so we wanted to do a more robust kind of analysis. And so what we did was we conducted a PCA using canopy cover, impervious surface, and housing density within 150 meters of the start of each transect to develop two scores that would be a measure of urban intensity and would allow us to look at things at a more fine scale. In our more robust analysis, we used a Bayesian model to determine the effects of urban intensity, habitat vegetation characteristics, and community composition on the stress levels of meadow voles and deer mice. And we have also controlled here for things that may affect baseline levels of stress-associated hormones, including sex, age, reproductive status, and season. Here we're looking at the results for the model for the meadow voles, and more specifically, the 95% confidence intervals for the coefficients in the model. So blue intervals indicated the variable had a positive relationship with stress-associated hormones, and red intervals indicated the variable had a negative relationship with stress-associated hormones. Any intervals that do not overlap with 0 are significant. In the meadow vole, we found that stress levels were not related to urban intensity, vegetation characteristics, or the community composition. We found that the stress levels, however, were related to body weight, sex, and reproductive status. So the first two variables here are the urban intensity scores. And as you can see, neither of these significantly affected the stress-associated hormone levels. Body weight, however, had a significantly positive relationship with stress-associated hormone levels. So this means that medovoles, which weighed more, had higher stress levels. Our next two variables are our measures of the habitat vegetation, and again, we don't see any significant effect of these. The next two variables we have here, deer mouse presence and the Shannon index, are the measures we use to kind of look at the community composition. So we didn't see that the presence of deer mouse had an effect on meadovole stress levels. The Shannon index, so basically a measure of the diversity of the small mammals at the site, likewise did not affect the stress levels. Next, we have temperature and month, which were controls for season. And then we have our controls for age, sex, and reproductive status, where we see that males had significantly lower levels of the stress-associated hormones, and particularly males which were scrotal. In deer mouse, we likewise did not see a significant relationship between urban intensity and stress-associated hormone levels, nor did any of the other variables significantly relate to the stress-associated hormone levels. Next, I have preliminary results of our analysis of tick burdens on the small mammals. For this, I did a zero-inflated linear regression model with the data from meadovoles and deer mice. So in this analysis, we see that tick burden was significantly positively related to the first urban intensity score. So this indicates that small mammals trapped in more urban areas had higher tick burdens. We also see that tick burdens were negatively associated with percent native vegetation, meaning that areas that had a higher percentage of native vegetation had lower tick burdens on the small mammals. Lastly, we saw that juvenile small mammals had significantly higher tick burdens than adults. So in summary, what we found was that small mammal communities differed significantly based on urban intensity, essentially the site type. We also found that small mammal stress-associated hormones were not significantly related to urban intensity, but were driven by other factors like body weight, sex, and reproductive status. And then we also found that small mammals have greater tick burdens in urban environments, and particularly sites with less native vegetation. Since we found higher tick burdens in small mammals in urban environments, this could be really important as small mammals act as reservoir hosts for zoonotic tick-borne pathogens. So this leads us to our next question we hope to answer, which is how does these higher tick burdens in urban environments affect Lyme disease dynamics? So Lyme disease is caused by a bacterial pathogen, Borrelia burgdorferi, and small mammals act as reservoir hosts for this pathogen, maintaining it in the environment and infecting black-legged ticks. Humans can be incidentally bitten by an infected black-legged tick, resulting in human cases of Lyme disease. Therefore, urbanization may have a role in modulating the prevalence of Borrelia burgdorferi infection in small mammals and Lyme disease risk of humans. Today, I'd like to acknowledge Matt Mulligan, who was the main designer of the study and did most of the work gathering the data and samples, Katie Fowler, who did the stress hormone EIAs, the many UWI interns who helped with the field work. I'd also like to acknowledge Seth Magley, Rachel Santimeyer, Maureen Murray, and Mason Fedino for their guidance and help with the design and analysis of this study. And of course, lastly, I'd like to acknowledge our funding sources, both the Davey Foundation and Grant Healthcare. If you have any questions, feel free to catch me at the Q&A session.